Our next research presentation of the day is from Jason Themanson. Jason is joining us to talk to us about neuroscience and specifically neural activity and in-game hitting. Jason.
Thank you, Scott. Appreciate it. Let me get the screen sharing going here. Is that visible to everybody?
Yes.
Perfect. Okay.
You can full screen if you like.
Let me do that. Better?
Better. Thank you.
All right. All right. Well, thank you again very much. So you're absolutely correct. Today I'm going to talk about neural activity and neuroscience at the plate. I'm going to talk specifically about neural activity we've collected in a lab and how it relates with actual in-game hitting performance. So hitting is, as everyone knows, a pretty hard but also dynamic psychological process. There's many factors going on throughout a plate appearance. There's obviously, first and foremost, pitch perception, seeing the pitch, recognizing the type of pitch, speed, direction, whether it's a ball or a strike, things of that nature. There's also the chess match between the hitter and the pitchers. There we go. Scouting reports, pitch types, what should I expect, what happened in the previous at bat. Similarly, there's learning the strike zone, whether it's a human umpire or now a robot umpire, the actual learning of the strike zone, pitches to stay away from, pitches to go after, decisions like that. Again, another psychological process ongoing all the time. And finally, there's within the hitter himself. There is game situation, expectations, what should I do, what do we need, what's the coach asking me to do. So all of these things are going on all the time. And many moons ago, research just looked at pitch perception, and we're going to circle back to that. But recently, we've been involved in examining the entirety of a plate appearance. So we've looked at pitch by pitch influences. We've looked at neural activity to the pitches themselves, as well as to the umpire's feedback between pitches. And what we've seen in our research that you can see at the bottom of the screen is we've seen that neural activity to pitches in this computerized paradigm I'm going to talk about today have been associated with task performance in this computerized task generally, as well as task performance following feedback. So after you get feedback from the umpire, how is your performance on the next pitch? We've also looked at neural activity to that feedback itself from the umpire and seen a relationship between, again, neural activity in the task with performance within the task. This has been well received and very interesting, but the question has always remained, how does this neural activity relate with the actual game? How does this relate with, does it translate to the field of play? Which is the thrust of the current study. So currently what we did is we had college baseball players, again, engage in our pitch perception task, and we measured their neural activity in this task. And when I say neural activity, I'm talking about event-related brain potentials, which I'll introduce here momentarily. And we wanted to see if that related with in-game hitting performance. So event-related brain activity is neural activity that's ongoing in your brain, time-locked to some specific event. In this case, a pitch. So this time point 0 here would be the initiation of that pitch event. In our paradigm, it's the release point, when the pitcher releases the pitch. Following that time point, you can see a pattern of neural activity associated with that event. And the way this methodology works is what you do is you repeat the event over and over and over again. In our paradigm, we had players look at 200 pitches to get the neural activity associated with that event and eliminate the neural activity that's ongoing in relation to other bodily processes, movements, etc. And from that, you get this event-related brain potential waveform. Within that waveform, there are multiple different components, as you can see here. The N2 we'll talk about more momentarily, that have been associated with cognitive functions and cognitive processes. So we're able to look in real time at what's going on within a player's brain, in this case, a hitter's brain, while he's viewing pitches, and look at these cognitive processes in real time objectively, not after the fact with some kind of question answer session. This is a real-time objective measure of neural activity. Again, for those of you who are unfamiliar, just a little demonstration of how neural activity is collected. a participant, in this case, a college baseball player, is seated in front of a computer monitor with a spandex cap on their head, and in that cap are sewn these sensors. So EEG technology is the same as EKG or EMG. We're just measuring the electrical activity coming from an individual. In this case, let me go back and play the video this time. In this case, there's an event on the computer, in this example, a red rubber ball, and at each point in time that event shows up, a marker is placed, and we can, from that marker, look at the neural activity immediately following that event and average all those trials together and look at the event-related neural activity as opposed to other electrical activity ongoing in the brain. So if we do this a number of times, we can pull out those individual components that are associated with cognitive processes. And one of them I mentioned before, the neural component that I mentioned previously is called, one of the neural components we're going to talk about is the N2. Now this N2 measured frontally or anteriorly from the brain has been associated with what's called response inhibition or response conflict processes. In essence, when you have multiple choices for how to respond to a stimuli, what do you stop yourself from doing? What response do you engage in? So in baseball, it fits perfectly. This is the swing take decision for a hitter. Should I swing? Should I not swing? Is that going to be a ball? Is that going to be a strike? Is that something I can hit, or is that something I should avoid? You have conflict between these decisions, and that process can be indexed through the amplitude of this N2 component. Another ERP that we're going to look at, another component of event-related brain potentials we're going to talk about today, is something called the medial frontal negativity. And again, negative is going up, so the more negative, the larger this component. This component's been associated with ongoing proactive cognitive control, engaging oneself through focus and actual cognitive engagement throughout a task. So being focused and maintaining focus throughout a plate appearance, pitch by pitch by pitch, staying in the task can be indexed through this MFN, medial frontal negativity component. So these are two components that we can see in relation to a stimulus, in this case, a pitch event happening, and we can measure them in real time in hitters while they're engaged in this task. So again, the point of this study was to see if this lab-based type of measurement, having players' neural activity measured in our lab, would translate to the field of play. Is there a relationship between these types of neural measures and their in-game hitting performance? And what is the nature of that relationship? What exactly is going on? So we move forward, kind of extending the lab and seeing if it translated to the real world. To do this, we had college baseball players participate in our study. They first came into the lab and had some measures collected, which I'll talk about momentarily, and we measured their neural activity during this pitch perception task that I'll describe in a few seconds. We had 9 players who had at least 50 plate appearances in the college baseball season following data collection, and then we expanded the analyses to include 12 players who had at least 100 plate appearances throughout their college career, mostly because of the numbers we saw were pretty surprising and pretty strong, and we wanted to be sure that the effects were real. So again, they came in and completed some individual difference measures on personality, anxiety, and affect, because these have been associated with either neural activity or task performance in other research. And then we had them complete our pitch decision task. And then we collected, after our lab collection was over and after the baseball season, we collected their publicly available hitting statistics, batting average, on-base percentage, OPS, and slugging percentage. And we'll focus primarily on OPS today, but the findings are similar across the different hitting metrics. In terms of analyses, we first conducted correlational analyses, and then what we did is, for any individual difference factor or variable that was associated with the hitting statistics, we included that in the first step of a regression analysis, and in the second step, we then included our measures of neural activity, and we examined R square as to assess goodness of fit of the models and to assess whether those patterns of neural activity had any unique relationship with hitting performance. So I've mentioned this task throughout, and I want to describe it now. This is the task that players engage in. They are simply seated in front of a computer screen, and they have a little response pad in their lap, and their job is to watch pitches from the viewpoint of being in the batter's box, and they need to respond whether a pitch is a ball or a strike. If it's a ball, they hit a button with their left thumb. If it's a strike, they hit a button with their right thumb. And we used VR software to try to make this as realistic as possible. Again, they are viewing the pitch from inside the batter's box. They need to respond while the pitch is in the air and make this ball strike distinction. Then after each pitch, they're given feedback from the umpire about their decision. And then another pitch shows up, and they do this repeatedly again so we can extract that event-related information. So here's a demonstration of the actual task. Again, oh, that did not work. Let's try this again. Apologize. Again, each pitch starts from the release point. They need to respond while the pitch is in the air. Immediately after the pitch, they get feedback from the umpire, and then the next pitch shows up. And we have this example is a right-handed pitcher throwing to a right-handed hitter, but we have it obviously with left-handed pitchers. We have it for switch hitters to make it as realistic as possible. Again, the time point 0, the event, is the pitch, and that starts at the release point. So we have repeated pitches, repeated feedback. In this study, what we're specifically looking at is their neural activity to the pitch itself and how that relates with actual college baseball in-game hitting performance. So looking at the neural activity just quickly to make sure that's actually there, this is a pattern of neural activity for all participants across all the pitches in the study. And again, what we see is that here's this N2 component reflecting response inhibition and response conflict processes. And here's this medial frontal negativity component, again, reflecting proactive control and engagement and self-regulation throughout a task. So we have the neural activity. Let's see if it relates with college actual baseball performance. So we first conducted zero-order, just bivariate correlations, and we were pretty astonished to see the magnitude of these correlations. So looking at these baseball statistics and these 9 players in the season immediately following our research, we found that neural activity, that N2 and medial frontal negativity that you just saw for each player, showed these massively large correlations with in-game hitting performance. And again, the more these are negative going components, so the larger they are, the more negative they are. So the larger these components are, the more positive or better their hitting performance. So for both the N2 and the medial frontal negativity, we saw these massive relationships with in-game hitting performance, suggesting that the more they engage in these response inhibition processes or these proactive control processes during task engagement, in our computer lab, the better their performance in the field of play. And to be honest, I was a little shocked by how large and strong these correlations were. So what I did is I reran all of the analyses using this larger sample of 12 players, looking at their career numbers. And again, the relationships were evident for both neural components across each of the different statistics. And like I said, I'll focus on OPS in relation to the regression analyses just to simplify the output. We also, again, looked at these individual difference factors just to, quality control, ensure that we weren't capturing variance measured in other ways. And as you could expect, so we have personality, anxiety, affect. We also collected players' ages, and we collected their career plate appearances, looking at career stats, assuming obviously the more plate appearances you have, the better a hitter you are, and you're going to keep being sent up to the plate. So as expected, we saw this very strong relationship between career plate appearances and these offensive measures. The more plate appearances you have, the better hitter you are. In college, it makes total sense. So looking at OPS specifically, we included career plate appearances, and we also included agreeableness, personality factor 2 here, agreeableness, which from the positive psychology literature and sports psychology literature, personality and affect make sense in relation to performance. The more agreeable you are, meaning the more kind of positive mindset you have, also the less amount of negative affect you have, again, you can see here, are associated with better performance. Specifically for OPS, therefore, we had agreeableness entered in our regression analysis along with career plate appearances in the first step of the analysis. And then the second step, we included the N2 or the medial frontal negativity to see the effect of those variables as well. Looking at the regression, Importantly, in the second step of the analysis, here we added N2 in the analysis, looking at its relationship with career OPS. And you can see here the overall variance accounted for in the entire model, very high. Also the change in R squared when we included N2, 0.18, suggesting the strong, unique relationship between neural activity and career OPS. The effects are even stronger and more kind of jarring when you look at the medial frontal negativity. Here again, the overall all squared, when we include all the variables in the model, 0.86, the change in R squared when we added medial frontal negativity in the second step, 27% of the variance, you could argue, is uniquely accounted for by medial frontal negativity activation in career OPS. Looking residually, just looking at the residuals of that relationship between medial frontal negativity and career OPS, with our 12 data points, we see this relationship. Again, the more negative medial frontal negativity, the higher or more positive career OPS. And this is the unique relationship residualized after taking into account career plate appearances and that personality factor of agreeableness. So we see this huge, in the world of psychology anyway, this huge relationship between neural activity measured in our lab and these in-game translationally real-world hitting metrics. And this is pretty exciting from our point of view. It has captured some interest. The one thing it made me think about, though, is there's a certain artificial aspect to having players come in and look at a computer and do this thing kind of in the, what if we made it more of a realistic experience? Well, that you cannot do that with event-related brain potentials because you need that very specific timing event marker. But one way you can get at neural activity measured objectively in a more real-world setting would be using just EEG and not looking at event-related brain components, but just look at ongoing EEG and neural activity typically measured through frequency bands. So I did that, and this is new, but it is a kind of exploratory that this analysis was just recently undertaken. So I looked at ongoing neural activity by frequency band, looking at EEG, and you might be familiar with this with sleep research. You've probably heard of alpha waves and beta waves and delta and things like that. But the interesting thing about EEG is you don't need a marker. You don't need an event. You don't need to repeat this event in the same way with such precision. You can just kind of hit record and measure neural activity throughout some engagement with a task. So in theory, what you could do is you could put someone in a EEG cap and have them looking at live pitches without the timing markers and just have them seeing real live pitches in vivo. Obviously, they're not moving, they're not swinging, you know, to allow for the capture of neural activity, but they could be standing in a real batter's box looking at real pitches in theory. So looking at this EEG analysis, I quickly ran some analyses and I found that there's delta band activity, which is very similar in terms of cognitive function with the medial frontal negativity. And there's theta band activity, which is very similar in terms of cognitive function in relation to the role that the N2 plays in conflict monitoring and response conflict. And I found a similar set of findings where greater frontal activity was associated with higher hitting performance, better hitting performance in games, and to a lesser degree, theta activity showed similar relationships. Looking at the correlation matrices, here we have it again, delta power, so more delta associated with better hitting performance, both following season and career, and similar but weaker with theta. So in conclusion, we show a translational relationship between these measures of neural activity collected in our lab during this task with in-game hitting performance. Specifically, looking at the event-related brain potential components, larger or more negative medial frontal negativity and more negative larger N2 amplitudes were associated with better in-game hitting performance, suggesting that these self-regulatory cognitive processes are potentially candidate mechanisms for enhancing performance outcomes in hitters. The more you are engaged in this response inhibition and self-control and proactive focus pitch by pitch by pitch might lead one to have better hitting performance. Similarly, looking at this time frequency analysis using EEG, where we don't need timing markers and the event precision that I discussed previously, we found similar effects with greater delta and theta power associated with better in-game hitting performance, which would, in theory, allow us to collect this neural activity from players while they're standing in a real batter's box looking at real pitches. Again, they're not swinging, they're not moving, but it's a more externally valid type of measure. So the implications for this, in my mind, are tremendous. We can objectively measure neural activity in real time while pitchers see pitches and see that it influences and relates with real life game performance. These measures index players' cognitive and psychological, self-regulatory and kind of focal attention control processes in real time without having to, well, what were you thinking? Well, what were you? So this can only provide an improvement in relation to the mental aspect of the game. This brings the measurement of psychological processes on par with the measurement of physical and physiological processes that we've heard about all day today, from sensors and force plates and high-speed cameras and markerless, biomechanics movements. That technology exists and is integrated into the industry for physical and physiological measures, and this would allow that to happen for psychological and cognitive measures. So again, this would create a more efficient, more effective communication for players with coaches across members of organizations to more effectively attack and approach and better the psychological or cognitive aspect of the game. Again, creating not only exploiting a data inefficiency, but also creating a competitive advantage, because these data don't exist. These data are not utilized by the industry yet. These data, as far as I can tell, people are not, capturing this event-related data as I've described here, and integrating it into scouting and development processes. So in the future, I would like to expand this research and actually look at collecting data while viewing live pitches in these hitters to see if the relationship persists across that environment. I'd like to look at players at different levels of play. Right now, this is obviously college players, but looking at players in minor league baseball, developmental or instructional leagues, single A, double A, and seeing how these relationships persist, looking longitudinally across seasons, across developmental arcs to see if this persists would be tremendous. And again, ideally, the way this would work is I would need access to many baseball players, and teams and organizations have many baseball players at their availability. So consulting or collaborating with a team to collect more data, to look at these questions, to look at neural activity during live pitches, or look across levels of play, not only would expand our understanding of this research and our relationships that we're seeing between neural activity and in-game performance, but also would be provide a data advantage to any teams. Again, this data doesn't exist out there, so teams would then have objective neural measures of their data to go along with the objective physiological measures and data points that they have for players to get a better picture and kind of exploit this market inefficiency and get a competitive advantage. Again, because these data do show this tremendously tight and strong relationship between neural activity and in-game hitting performance. With that, I'd like to acknowledge Illinois Wesleyan and Illinois Wesleyan's baseball team for working with me on this research over the years, my students and colleagues for helping make the research better and making it actually work, my family and for putting up with me, SABR for this opportunity to present with all of you today, and all of you for listening. So with that, I will head to the questions. Joe asked, what are the sensors we're using? The sensors we're using are silver chloride. So they are silver chloride sensors sewn in the cap. They also can then, we do them to measure blink activation as well, because blinks are tremendously strong neural signatures, because the blink muscle is on the outside of the skull, whereas the brain is under the skull. So you have to account for blinks when you're measuring neural activity. So we're using silver chloride sensors. The samples ask if the sample of 9 players is a separate group of players. No, No, the 9-player sample was included within the 12-player sample. So there is redundancy there. Absolutely, that is not a separate group of players. It's just that I was just floored by the size of the correlations with the 9, so I wanted to see if we could make it more robust and figure out a way to include more players and also more plate appearances for the statistics. So I expanded the same sample to include the 12 players. Richard asked, would there be any difference in a player's response to a decision conveyed by a human or robotic umpire? I would probably argue not. Even though a robotic umpire's strike zone might be more consistent game by game by game, you still are going to have instances where players are thinking that's a ball and it's going to be called a strike. And we've heard anecdotally from people last year that dealt with robot umpires. I mean, that happens. So it's more, self-regulation is more like, when you find out you're wrong, what do you do? When you find out you're right, how do you respond? And kind of this learning mechanism. So that wouldn't matter human or robot umpire. David asked, did you correct for career statistics entering the season? Did the exercises improve performance or only indicate the likelihood to be successful? So we did correct for career statistics entering the test season in that initial analysis. And then obviously we just expanded to the career statistics in the career analysis. So there's no correction needed. And that finding was still very robust. The exercise, we don't have data yet on longitudinally whether it improved performance. This baseball season, we'll be able to see players that were in our study last year to see if maybe their participation in the exercise, but we haven't really given them feedback quite yet. This task, this capability to exert control is malleable. It is trainable. There's a long history of the Olympics using biofeedback in marksman, which dates back 30 years now. So there is an ability to, and an implication that you can use this to improve performance, but we don't have that data yet for this study. Do you think any teams are using analysis like this? No, I do not think there are. There was a company a few years ago that did something with when you can recognize pitches, and Mookie Betts is a very famous case example of that. But that's more a very perceptual process and not a cognitive process. And I don't know if they still do what they did years ago. So I don't think that, as far as I know, there are no major league teams or college teams using this analysis. I'm sharing the data with the team that I collaborate with, which is Illinois Wesleyan. As part of giving me access to the players, I give them the data that I collect and my write-up of it. But as far as I know, no one's taking advantage of this. Christian asked, can a player improve in these areas? Yes, you can improve. This is a cognitive process. So with training and with practice, you can improve your ability. In essence, we're talking about your ability to focus and your ability to control and regulate performance to meet your goals. So as I mentioned before, the Olympics have used in marksmen, they've used biofeedback to train them to have a pattern of EEG, which is relaxed concentration versus agitated, because you want to be relaxed to be able to have quiet eye and quiet trigger fingers, and things like that for marksmen. Joey asks, what are researched ways to change or improve N2? And there are some sport-specific studies, not with baseball, but it's just practice. And with any cognitive training paradigm, it's targeting specific processes that are related with the task at hand. So you can target, in this case, response inhibition processes to make someone more effective at stopping something they want to do, like swing. And as I'm sure a baseball fan, you've heard that a lot of players, every pitch they start their swing, and then they decide that they need to stop or continue the swing. That's what this N2 kind of process mimics. So you can, just through practice and focused concentration on that response inhibition and stopping, you can get better. Daniel asked, do you think such results could correlate to defensive performance as well? That I'm not sure of. That would be interesting, though. I mean, the one thing about this is with this we could look at a batted ball in play. I'm thinking like in terms of we need a timing marker for the event-related brain potentials. You could look at a timing marker and measure reflexive energy. I don't know cognitively though what you are having to maybe inhibit or control. I guess route, you could look at maybe route to a batted ball would be something that you need to control or exert control over throughout the process, like hitting. But that I haven't thought about that. That would be interesting. Jason asked, from a scouting perspective, would adding neural activity as a trait to a player's profile be useful? I would caution against comparing players to players necessarily, even though I talk about it today. This data is very individualized. I mean, it could be added, I'm assuming, but I would be very hesitant to say that because a player has a larger N2, that player is going to be a more effective hitter, just because there's so many other variables in play, right? Just because a player is stronger doesn't make them a better pitcher. So I think there's a place for it. I really do think there's a market inefficiency here where this data should be included. I just don't know to what degree or to what end you would want that data to be part of. But having it as part of a larger profile, absolutely, I think would be valuable. Masahiro asked, do you think there's a possibility that preparation before the pitch, like expecting pitch type or narrowing their own choice, would help? I absolutely do. I think, that's kind of what the medial frontal negativity index is this ongoing, continued type of proactive control. So that is exactly what we're getting at, like this mental specificity and control and preparation for the task is exactly what we're going with here. With that, I think my time is up. So I appreciate everyone paying attention, and feel free to send any more questions my way if you like. I appreciate it. Thank you very much.